Welcome to Ash Jottings. Today we are going to talk about DNA, that is deoxyribonucleic acid, its chemical composition. DNA is a self-replicating material that's in every living organism. It's one long molecule that contains our genetic code or recipe. This recipe is a starting point for our development, but DNA's interaction with outside influences such as lifestyle, environment, and nutritional ultimately form the human beings. While most DNA is found in the nucleus of cell, some amount of DNA can be seen in the mitochondria. Let me just use a pen. Okay. Perhaps the most fascinating part of the process is the fact that nearly every cell in our body has the same DNA. So we know that DNA contains genetic instructions for the development and proper functioning of all living organisms. All living things contain DNA genomes. While a possible exception is a group of viruses that have RNA genome. But um, these viruses are not normally considered as living organisms. The main role of DNA in the cell is a long-term storage and information. We know that when it is connected to the genetic right? Sometimes a genetic code. So it is long-term storage of information. The genome is often compared to a set of blueprints since it contains the instruction to construct other components of the cell, such as proteins and RNA molecules. Now, the DNA segment can carry this genetic information, are called genes, and there are DNA sequences that have structural purpose or are involved in regulating the expression of genetic information by acting as deoxyribonuclease. Right, so that is an introduction about DNA. Let's move on to the next slide. As I have mentioned, there are types of DNA which is present. Let's just talk about it. So nuclear DNA and mitochondrial DNA. So nuclear DNA, it is present in the nucleus. Well, so the mitochondrial DNA, it will be present in the mitochondria. Now, usually in nuclear DNA has two copies per cell, whereas here, if you can see, it has 100 to 1,000 copies per cell. The nuclear DNA structure of the chromosomes is linear with open ends and include around 46 chromosomes containing 3 billion nucleotides. In the mitochondria, you can see the chromosomes are usually closed. They are circular structure and they contain, for example, 16,569 nucleotides in human. Nuclear DNA is diploid, ordinarily inheriting DNA from both the parents, and the mutation rate is very less, around 0.3%. The mitochondrial DNA, it is haploid and it is only coming from mother. The mutation rate is comparatively greater or higher than the nuclear DNA. So Apart from this nuclear DNA and mitochondrial DNA, there is also another form which is called the chloroplast DNA, or it is written as ctDNA. Like mitochondria, this chloroplast also possesses double-stranded circular DNA. So it has double-stranded circular DNA, right? It is represented by several copies that lies in the matrix, and the functioning of the ctDNA is controlled by the nuclear DNA. So these are the types which are available, and there is also another type of DNA which is interesting to know, that is the catalytic catalytic DNA. These are also known as the deoxyribozymes. Now, this deoxyribozymes, like if you see in all living organism, except few viruses, DNA is exclusively used to store genetic information and has never been expected to serve as an enzyme. Now, enzymes are mainly proteins, we all know that, but there are some RNAs which has a cleaving function and it acts as an enzyme. Now, however, a single-stranded DNA molecules are found to have this catalytic action, single-stranded DNA. Let me just mention it here. So few single-stranded DNA have this catalytic action function, and to greatly accelerate the transformation in a substrate, a variety of chemical reactions which are occurring in the cells, like you can just say, let me just write it down here, cleavage, cleavage, ligation of DNA, RNA, or you can say in the 5' phosphorylation, then in 5' adenylation, uh, ADEN, yeah, adenylation of DNA, then in the thymine repair, thymine dimer repair, and also in DNA cloning, DNAs are found to catalyze cleaving of RNA molecules using certain cofactors like histidine. So the single-stranded DNA molecule acting as a deoxyribozymes form um, form an intricate structure by forming a binding pockets for small molecules, which actually enhances the activity. That is, they form small pockets. Okay, the small pockets, so which helps in their function. Okay, so that is all about the types of DNA. You have the nuclear DNA, which you're very familiar with. Then you have DNA which is present in the mitochondria, mitochondrial DNA present in the chloroplast, which is also known as ctDNA. And then you have the catalytic DNA, which is a single-stranded DNA and which can actually help in cleavage or acceleration, transformation in a substrates, like variety of cleavage, ligation, 5' phosphorylation, 5' adenylation, thymine dimer, okay, so and so. So they will be present as the structures will be present as a forming of a binding pockets for small molecules, so that it will enhance the catalytic activity. Now let's move on with the next slide. Now let's see what is DNA made of. As you can see, the DNA is made up of a molecule known as nucleotides. Right? Now, let's see what is this nucleotides made up of. Nucleotide consists of sugars, phosphate, nitrogenous bases. And when you talk about nitrogenous bases, you have adenine represented by A, cytosine, C, guanine, G, and thymine T. Right? Now, the sugars, when you talk about the sugars of deoxyribonucleic acid, okay, let's write it down here. The sugar structure, the sugar, okay, you have H, H, you have H, OH, I'll just show you. Now, this is the structure of ribose sugar, which is present in RNA, okay? Which is present in the RNA. This is a ribose sugar. The difference between these two is that DNA sugar is deoxyribose. Deoxyribose, okay? Now, DNA contains this ribose sugar, which lacks a hydroxyl group on the second carbon. So it does not have this. When you actually remove this, that will be your deoxyribose. Okay. Now, so you get it. This is the RNA, the ribose, which has two hydroxyl group. But once you remove one group of hydroxyl, okay, then this will be your deoxyribose. Okay. Now, absence of this OH group will help to stabilize the structure, which helps to stabilize the structure of DNA and making it more resistance to hydrolysis. Okay, so the sugar which DNA has is deoxyribose. That means it will not have a hydroxyl group, okay, which actually helps to stabilize the structure of the DNA and also helps in the resistance to hydrolysis. Now, next is when I talk about, okay, let me just take now, as I had told you, the sugar, let me just draw it here, more clear. H, you have the H, and you have the CH2OH, right? This is the deoxyribose sugar. Now, the numbering will be 1, 2, 3, 4. And this is if I count which is present, okay? Now, let's talk how it is going to form a phosphodiester bond. Okay, now the sugars of these nucleic acids are converted to another sugar, oh sorry, not converted, it is connected to another by 3 dash, let's write it down here, 3 dash to 5, 3 prime to 5 prime phosphodiester bond, or you can say linkages. That is, it is going to form, that is sugar bond to phosphate sugar. It will be like this. So in one side, the sugar will be present, or it is attached to the phosphate. Okay, from, if you see this, from here it is connected to the phosphate. Yes, double bond on, you have, okay, now this will be connected with the, to the fifth position of the sugar. Okay, that is the connected to the CH2. Okay, this is how it will be bond, that is the two sugars will be bond to the phosphate group. Okay, now the chain, the OH group, that is, I have mentioned it here, the OH group of third carbon of one sugar is connected to the OH group of fifth carbon of an adjacent sugar. In a phosphate group. Now, this chain of repeating sugar phosphate unit makes up the backbone of the, so this is what makes the backbone of nucleic acid. And this backbone remains constant and unchanged. I get it? So we learned that the nucleotide are composed of sugar, phosphoric acid, and nitrogenous base. So we are talking about sugar. The sugar mentioned is a deoxyribose sugar. So the difference between the ribose and the deoxyribose is that in the place of second carbon OH, it will be only the OH is not present. It is only the H which is present. Okay. Now, how it is going to form a phosphodiester linkage, that is how it is going to connect with another sugar which will act as a backbone of nucleic acid, which cannot be changed, or it is constant. That is, it is going to form OH group of the third carbon, OH group of the third carbon of hydroxyl group of the third carbon of one sugar is connected to the hydroxyl group of the fifth carbon of the adjacent. So it is going to connect to this part. So if you see from here, you'll have again a connection coming from this part, sugar in the phosphate group. So the two sugars are connected by whom? By the phosphate group, right? Now, that is why it is called diester. There is two link diester linkages which is occurring here. Okay, so this is the backbone of nucleic acid, right? Now let's talk about, just erase this. Now let's talk about the phosphate group, phosphate. The phosphate group contains what charge? It is negative charge. Now this means in aqueous environment, this hydrophilic so it will have a hydrophilic region So this is the part which is going to interact with the water, right? It allows water and interact with the polar molecule to stabilize the structure of the DNA. So this helps to stabilize the structure of DNA. Then the phosphodiester linkage are much less likely to be attached on by the nucleophilic. Also the attacked by the nucleophilic, because it's very consistent. Nucleophilic agent that is less susceptible to hydrolysis. Okay, right? So that's all about phosphate group. So phosphate group, you can just say it is negatively charged. It has got a hydrophilic. That is, if you see the structure, you know that in the double helical structure of the DNA, if you can see this, right, this part here, this side part is where the sugar and the phosphate is present, and inside you have the nitrogenous bases. So this side, it is hydrophilic, and the inner side will, the nitrogen bases are, they will be hydrophobic, which doesn't like water. So this actually helps, or it actually is one of the point which actually helps in the stabilizing the DNA double helical structure. And also it actually is, there is a chances of less chance that the nucleophilic agent cannot attack of nucleophilic agents is less likely to take place. That is nothing but it is less susceptible to hydrolysis. Okay, now let's go back to the, okay, so we have seen what is sugar, and now let's talk about the nitrogenous bases. Okay, so we know the backbone does not change, right? As I mentioned it, the base vary from one nucleotide to other. So there are two nucleotides which are present, which you know is the purine and the pyrimidine. So let's talk about pyrimidine first. Okay, so this is, sorry, let me just selective color. Let's talk about stuff. Pyrimidine. Pyrimidine is formed of one ring, right? It is formed of one ring. And the ring is what? It is hexagonal. Hexagonal, or you can say heterocyclic. Heterocyclic. And formed of 4 carbon and 2 nitrogen atoms, right? This is a single ring, it's hexagonal and heterocyclic, and it is made up of 4 carbon, 4. So you can see from here, 1, 2, 1, 2, sorry, 1, it's nitrogen, let me over 1, 2, 3, 4. Okay. Then the nitrogen atoms are positioned and the first and, sorry, this is the position. First and third position, they are nitrogen atoms of position in the first and third. And rest of the position are occupied by carbon. So you have 1, 2, 3, 4 carbon. C, 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 C here. And the rest 2 positions, that is in the first and in the third position, you have nitrogen. Now, pyrimidine nitrogen bases are found in DNA in the form of cytosine, thymine, or if it is RNA, you have uracil in the place of thymine, right? So now that you know how is the structure, please try to connect everything when you learn the structure about, okay, how it is going to be. So if you see cytosine, all the three nitrogen bases, all the three nitrogenous bases, the carbon atoms are present at the second position, has an oxygen, right? If you see, you can see the numbering is 1, 2, 3. So the whole thing like 1 and 2, 3, it is like that for whole thing. So if you see it is the second carbon, you can see each of them will have a oxygen, right, at the second carbon. So let's put it like this: second carbon position has an oxygen attached, right? And by double bond, right? So second one is double bond O, which is present. Okay, 1 point, how to remember the structure. Next, the nitrogenous bases. So the three nitrogenous bases differ in the functional group present on carbon 5 and the presence of C double bond. Okay, you can say C double bond or CnH2 group at the carbon 4. So you can see the where here it is similar. If you see all the C2 carbon position, they have a double bond O here. They differ in the C5 carbon atom and the presence of a C5 atom where the double bond O is present. Where is it present? It is present here in the uracil and the thymine, right? So just know, remember it as a thymine and uracil. And you have a CnH2 at carbon 4. At carbon 4 you have, that is the third point you have to remember, you have the NH2, right? So pyrimidine, as I mentioned, it's a one-ringed hexagonal heterocyclic. It consists of 4 carbon and 2 nitrogen. And the forms of pyrimidine is cytosine, you have thymine and uracil. In the place of thymine, uracil is present in RNA. In order to remember the structure, please, I have mentioned 3 points here. You just have to remember that although it is a single ring, remember that, and the carbon 2 will always be having double bond oxygen, right? Now, the difference of the functional groups where it differs is at the carbon 5, okay, carbon 5, and sorry, I think I messed it up. Okay, carbon 5 and the presence of, yeah, double bond. So carbon 5 here, okay, I think I messed it up here. So it is like, you cannot see. Sorry, let me just write it here. Differs groups present on C5. The differ is in the C5. Okay, the second point where it is differ. If you see in cytosine that the 5 position, it is like this 5, right? Nothing is present there. But in the uracil also it is not present. But if you talk about the thymine, you have CH3 there. Okay. So this is your second point. And the third point is you have, see in the fifth, that is the fourth carbon, let's talk about C4. What is that you have? You have either a double bond O, okay, or you have the NH2 group, right? Yes, now it's clear. I'm sorry for this. Okay, now it is clear. Now you can just remember pyrimidine like the way I just told you that it is a hexagonal. Then just remember how many carbon atoms it has, how many nitrogen atoms. Then the numbering also you should know, because based on the numbering only we are going to say the difference between all those things. So cytosine, thymine, and uracil, common thing is the at the carbon 2 position you have double bond O, and in again the difference lies in the C5 carbon and C4. So if C5 has CH3, okay, then it is called thymine, and C4 where it will have double bond in the case of uracil and thymine, whereas cytosine will have NH2. Okay, now talking about, so you can just say cytosine as 2-oxy, right? 4-amino pyrimidine. Thymine as 5-methyl. Then the rest part is uracil, so it is a uracil. For uracil, you can say you have 2 bonds, right? So dioxy pyrimidine. Okay. Now let's talk about the purine. Purine is a 2-ring nitrogenous compound which is dicyclic, and 1 pyrimidine ring and 6 atoms. So you have 1 ring and you have 2 ring. This is the pyrimidine ring. Okay, and this ring is known as imidazole. Imidazole ring. Okay. Now, the atoms are numbered anti-clockwise in the pyrimidine. So if you take this is a pyrimidine part, right? The next thing is it is in anti-clockwise direction, the numbering 1, 2, 3, 4, 5, 6. And here it is clockwise, and 7, 8, 9. So it is clockwise and that is anti-clockwise. Okay. Then it is 2-ring and it is dicyclic. I'll just write it, dicyclic. Okay, it is dicyclic, 2-ring structure with the purine ring and imidazole and the other part. And purine is anti-clockwise, whereas imidazole part is clockwise. While you number it, you have to remember these points. Now the 2 rings share carbon atom present in the 4th and the 5th position, right? They share the same carbon. The nitrogen atom in the purine ring are in the R4, right? You can see 1, 2, 3, and 4, right? And present at the 1st position, you have 1 nitrogen. Okay, now you have 2nd nitrogen present in the 3rd position. Then you have the 7th and 9th position you have. So you have guanine. The 2 purines are guanine and adenine, right? So it's a you just the difference lies here. You can see it's a double bond, and here it is an amino group, and the absence of amino group which is not present in this position of carbon 2. So you can say like the difference, you can say that bonds, you have a double bond present here, rest of the things are the same. Only thing is the position 6, carbon 6, okay, if it has double bond O, then it is guanine, okay. Let me just come back again to that point. And if it is, you have an amino group, it will be adenine with an extra bond at the nitrogen 1, right? Then apart from this, you also have a amino group in the position 2, C2. You have one more NH2 group here, right? Then it is guanine. So remember it. This position is 6 carbon. So sixth carbon, if it is double bond O, then you remember it guanine, and it is adenine in the place. So if it is adenine, it just have another NH2 group. So guanine does have an NH2 group, but it is present in the second carbon. Okay, so that's all about the chemical structure of DNA. Thank you for watching.